Hello everyone. Today we are going to do estimation of hardness in given water sample. So I have already told you about the basic principle of this method. So we are going to estimate with the help of EDTA, that is ethylene diamine tetraacetic acid. So let me give you a live demo for this. For all the three titrations we need burette, conical flask, funnel, pipette and reagents required for this titration. We need a burette solution, that is EDTA for all the three titrations. Then we need for the first titration standard hard water. For the second titration we need water sample, and for the third titration boiled water sample. For all these three titrations we need buffer and indicator, which is EBT, and we also need wash bottle. So the first titration is standardization of EDTA. So what we do that, previously washed and rinsed burette we will take to this, we will keep funnel on it and then add burette solution into it, right. So we have to keep slowly, and then after that we have to remove funnel, and then since it is above the 0 we have to bring it to the 0. Since it's a colorless solution, we have to see the lower meniscus of the solution. Okay. So after that we will fit it into the clamp. Then for the first titration, so we need a standard hard water. For this we will take conical flask. To this we will pipette out 20 ml of the standard hard water and put it into the previously rinsed conical flask, right. Now as you know that the last drop of the pipette solution we should not blow it, we have to just give a small jerk. So last drop must be inside the conical flask and then place it back to the T1 reagent bottle. Okay. So we have pipette out 20 ml of the standard hard water. Since this experiment is carried out at one particular pH, that is around 10, so we have to add ammonia buffer. For this we will add one test tube full of ammonia buffer. So we will pour into the conical flask. To this we have to add 3 drops of the EBT indicator, 1, 2, 3. Then we have to swirl it a bit. Now you can see that it becomes wine red in color. So when it shows that calcium ions are present in the given standard hard water, which we know that prepared by calcium carbonate, right. So now this is a wine red metastable complex which we have to titrate against EDTA taken in the burette. So this wine red color solution going to titrate against EDTA, right. So we keep adding EDTA solution from the burette with a constant swirling, and then we will focus on the color. So before we reach to the steel blue color, you can see that it will, and one intermediate color will come. So focus on the color slowly and then keeps adding EDTA from the burette solution. So you can see that still wine red color is there. So now you can see small changes started appearing. Slow down. So this is your bluish purple color. So once you get this color you have to be very careful, and then we have to add slowly 1 drop. If you add, it will become steel blue in color. Can you see? You have to close it properly. There should not be any dripping from the burette solution. So this is the end point of the titration. So after adding EBT and buffer, you could see that it was wine red color, right. Then we titrated against EDTA, then the end color is steel blue, but just reaching before the steel blue there is an intermediate color which is bluish purple in color. right. So, this is the transition 1 to 2 and finally the end point is the steel blue color. Once you get this steel blue color, you will stop the burette as I said. Now, you will take the burette in hand and note down the value, right. So, that will be your V1 ml. Now, also you will make sure that you must get a concordant value. Concordant value means you must get 2 consecutive reading must be same. So, once you get that, physically you have to get it attested after recording in your observation notebook. So, for the second titration, again you take the burette in hand and remaining whatever volume is remaining to fill up to the 0, again you will place the funnel and then add EDTA into it and make sure that it should be slightly above the 0, and then if there is an excess of EDTA in the burette, just bring it up to the 0 so that lower meniscus must rest on 0 mark. Also you make sure that there should not be any air bubble trapped in the nozzle part of the burette, right. So, this is then put it back into the burette stand. So, again you take conical flask. In that conical flask you will add pipette out 20 ml of the given water sample and then add 1 test tube of buffer and 3 drops of the indicator. So, as soon as you add 3 drops of the indicator, it turns to wine red color, right. This wine red color you will start titrate against the EDTA solution which is taken in the burette, right. Before you reach to the end point of the titration, you can see that it gets turned into the bluish purple color, right. So, the experiment remains same, only you are changing your pipette solution, okay. So, wine red to a purple to the steel blue color. Again you pour it off, again pipette out for titration 3 where we estimate the permanent hardness present in the given water sample. So, 20 ml again you pipette out in a conical flask, you will add buffer, 1 test tube, 3 drops of indicator, again titrate it against the EDTA taken in the burette, wine red to the purple and then to the steel blue color. Once you get steel blue color, you have to stop it. Make sure that for each titration, you must get a concordant value. With this, we finish our experimental